Jenny Lee,
Sadie Wong
He
presents Diffusion and Osmosis Lab. Lab 1. Diffusion osmosis through a membrane. Purpose: to compare the water potential for different pairs of solutions by modeling a cell membrane with dialysis tubing. To understand the real-life implementations of this effect, let's look at cells under the influence of intravenous injections. The ideal situation would be an isotonic solution where the cell and the IV have the same concentration of solutes in water. Hypertonic solutions with too much salt will draw out too much water and lead the cells to shrink and die. This is called exosmosis. IVs with pure water are hypotonic, and water would flow into the cell. This is called endosmosis, which can lead to cytolysis. How would you determine the best concentration of solutes to give a person before you introduce them to the IV bag? Conduct the red blood cell study similar to the one Hamburger conducted in 1882. Draw out the person's blood and determine the perfect concentration of solutes so the environment is isotonic. Hypothesis: solutions with high water potential will allow water to diffuse into solutions with lower water potentials. Materials used. Procedure: one pair of solutions were chosen. A cell was modeled by using dialysis tubing and tied off at one end using floss. A graduated cylinder was used and 10 milliliters of the first solution was poured into the dialysis tubing. The other end of the tubing was tied off. The mass of the cell was masked and recorded. The cell was placed into a properly labeled styrofoam cup and the second solution was poured until the entire cell was covered. After 13 minutes, the mass of the cell was masked and recorded. Observations were recorded. Step 1 to 6 were repeated for 3 different pairs of solutions. The following solution combinations were used: sucrose and sodium chloride, starch and sucrose, sodium chloride and starch, water and water. Repetition was done simultaneously in order to save time. Table 1.1. Solution pairings and masses. Our prediction for the first pairing is that the mass of the dialysis tubing would decrease, the second would decrease, the third would increase, and the control would be the same. Table 1.2. The percentage change in mass was calculated. The mass of the first pairing and third increased, that of the second decreased, that of the control remained the same. Observations: there were liquids on the electronic scale. All solutions were clear except sucrose, which was sticky and slightly yellow. The dialysis tubing had air bubbles, different amounts of solutions per cell, and different shapes.
Discussion and analysis. Factors that determine the rate and direction of diffusion. Passive transport, down the concentration gradient, from high concentration to low concentration. Molecular weight, smaller and lighter diffuse faster than heavier and larger. Surface area and volume ratio, the higher the ratio, the faster the rate of diffusion. However, it cannot be overly large because it would not be able to sustain life. Purity, the more pure the solution, the faster the rate of diffusion. Factors that determine the rate and direction of osmosis, the diffusion of water. Temperature, high temp equals more kinetic energy and then the faster the particles will move. It moves up the concentration gradient, from low to high. Distance, long distance equals longer time, shorter distance equals shorter time. The following is the solute potential calculations for the solutions used. Evaluation of prediction for each pairing. Our prediction was correct for everything except for the first pair, sucrose and NaCl. The mass change has everything to do with water's potential. Water potential is water's capability to do work. Solute potential. Solute potential is the measure of change in the water potential of a system due to the presence of solute molecules. The more solutes there are, the less water potential is present. Water wants to travel from a high solute potential to low solute potential. The lower the solute potential, the more the water diffuses. The movement of water molecules for the first pairing, sucrose and NaCl. Sucrose has a high water solute potential and therefore water would diffuse towards the outside, making the cell lighter. The movement of water molecules for starch and sucrose. Because starch and sucrose has equal solute potential, the movement of water molecules would be minimal. The movement of water molecules for NaCl and starch. The water molecule would move from the outside, from starch to the inside with NaCl, making the cell heavier. There will be no change for the movement of water molecules in water. Dialysis tubing is different from a cell membrane. Both have a semi-permeable membrane, but dialysis tubing is one thin layer of cellulose. A cell membrane has lipid bilayer for a membrane. Dialysis tubing transportation is limited to osmosis and diffusion. Cell membrane uses proteins and enzymes. Transportation includes simple diffusion, facilitated diffusion, phagocytosis. Glucose, broken down by starch, is transported by the blood through the body to all cells in most animals. Glucose is a monomer and is smaller than starch molecules, easy to diffuse through membranes. Passive transport requires less ATP, saving energy for the cell. Glucose is needed to create ATP.
created using Powtoon.